Hi friends, today I have come across with a concept called regulation of gene expression. We here observe two terminology, gene expression and regulation. The gene is expressing, that means it is going to synthesize the polypeptides. At the same time, it is going to be controlled. So that is considered as regulation of gene expression. In order to know about this process, I have taken an example of lac operon concept. Lac operon is a very good example for regulation of gene expression. We can study how a bacterial cell is going to control the process of expression of the enzymes in the presence or in the absence of the substrate. In order to study this whole process, first we should need to know about the basic idea of this operon system. So here you can see a diagrammatic representation. The whole thing is considered as a E. coli bacterial cell. The blue color ones are the lactoses. If lactose enters into the bacterial cell, the E. coli has to synthesize, that is nothing but gene expression, the production of beta-galactosidase. When beta-galactosidase is synthesized, it going to break down lactose into glucose and galactose. And this glucose and galactose is used for the metabolic activity of the E. coli cell. This is a one scenario. Suppose if the lactose is not present, then how the genes are going to be expressed? Whether is it going to express or it's not going to be expressed is the today's topic. The principle that is lying behind is the same what I said. In the presence of lactose, in the absence of lactose, what is the fate of the genes that are present inside the lac operon? This is the operon. So what does the term operon refers to? Operon is nothing but coordinated regulation of expression of several set of genes. Operon is not a single gene. Two or more genes has to club together. They have to work together in order to bring out a particular activity. The operon is divided into two parts. One is regulatory gene, another one is structural gene. Regulatory gene is nothing but inhibitor gene. It produces a protein called repressor protein. This plays a major role in the presence as well as in the absence of the substrate. Structural genes, there are three structural genes Z, Y, A. All these three, they work together one after the other. Once again, this depends upon the presence or absence of the substrate. Regulator is a controlling region. It produces a protein called repressor protein which binds to operator and controls the gene expression whenever it is required. Now we will move on to the actual concept lac operon. Francisco Jacob, a geneticist, as well as Jacques Monod, a biochemist, they both of them work together in this lac operon concept. This lac operon is a polycistronic. You might have all heard about a monocistronic and polycistronic gene. Monocistronic means produces only one kind of a polypeptide that's going to carry out only one function. Monocistronic is the one functioning gene, but polycistronic gene is having two or more sets of genes that work together and bring out one particular process. That's called polycistronic gene. Like a lac operon, even we have other type of operons like tryptone operon is called tripoperon, histone operon is called histoperon, valine operon is called valine operon. So in lactose, we have a I that refers to inhibitor that codes for repressor protein. Z gene codes for beta-galactosidase which hydrolyzes lactose into glucose and galactose. Y codes for permease that helps in the permeability of lactose into the cell. A gene codes for transacetylase. For the overall process to carry out, we call lactose as a substrate or inducer. In the presence of the lactose, we call the operon in switch on condition. In the absence of lactose, we call it as switch off condition. First, we'll know what is the term switch off refers to here. If operon is operating, it's called switch on. If operon is not operating, it's not transcripting as well as transcribing, it's not synthesizing any of the genes that are responsible for the metabolism of the lactose, then we call it as switch off condition. Whether lactose is present or absent, inside the bacterial cell, that is E. coli cell, always there will be a minimal lactose operon activity will be taking place throughout its day or throughout its lifetime, because lactose will be operating in a minimal quantity in order to transfer the additional lactose that are present in the nutritional media. If the operon is not activating, then it cannot able to transport those additional lactose from the nutrient media into the bacterial cell. Here you can see the operon has started its function. There is no appearance of a lactose in the medium. The inhibitor has produced the mRNA. This process is called transcription. It has underwent transcribe, that is translation, produced the repressor protein. The repressor protein directly goes and binds to the operator. Promoter is the region where it's going to initiate the process. Since the operator is blocked, the promoter cannot initiate the process of transcription. Therefore, there is no production of mRNA and there is no synthesis of enzyme. Therefore, we call this whole process as switch off condition. When you see switch on condition, there is a presence of lactose in the nutrient media. The inhibitor has produced the repressor protein. The repressor protein has binded with the lactose. Now this area is set free. The promoter initiates the process of transcription. There is a synthesis of mRNA from these enzymes. Now they carry out a translation and produces the enzymes beta galactosidase, permease and trans-acetylase. And now first the permease comes into the action. It brings all the lactose present in the nutrient media inside into the bacterial cell, like how we observed here. Once lactose enters into the bacterial cell, we can see the beta galactosidase comes into the action and break down the lactose into glucose and galactose. Okay. So after this, the glucose and galactose are utilized by the cells. So here in both the nutrient media, you can make out the difference. The green color ones are the lactose, the black color ones are the bacterial cell. The bacterial cell are minimum in their number since there is no lactose appearance in the nutrient media. If lactose is present in the nutrient media, it has utilized the lactose maximum and it has underwent the replication or a reproduction, we can say, and it has increased its colony. This is how a small bacterial cell carry out its gene expression. Whenever it is required, it's going to initiate the gene expression, and whenever it is not required, it is going to inhibit the gene expression. Therefore, overall we call this as regulation of gene expression. So, if you have gone through the topic, if you have understood, please subscribe as well as like. If there is any comments you need to share it, you can put your comments as well as share to others also. Thank you.